So welcome back again. I'm your mentor Dr. Silendra, and we are solving IIT JAM 2010 paper under Vigyana. So we'll understand the nature of question, we know the level of difficulties, and we will know why certain options are correct, why certain options are wrong. For example, here in the question 36 you can see a bacterial culture that is growing at 20 degrees centigrade was shifted to 45 degrees centigrade, and incorporation of which of the following fatty acid in the membrane will be beneficial for its survival. So this level of difficulty is 3. Alright, and you must be aware of the function of fatty acid in the membrane. Okay, so the first option given is long chain fatty acid must be there, short chain fatty acid must be there, unsaturated fatty acid must be there, and saturated fatty acid must be there. So they are asking which kind of fatty acid in the membrane will be beneficial for its survival. So it must be long chain. This is because short chain fatty acid will not be much helpful because it's related to melting temperature, as well as because you are increasing the temperature from 20 degrees centigrade to 45 degrees centigrade. So if unsaturated fatty acid is there, unsaturated fatty acid already they are liquid in nature, right? So they will melt, they will not be much beneficial, though it must be saturated, right? That is solid and not going to melt easily, right? And they must be not short chain, they must always be the long chain. So this option you can do it if you know, if you have an idea a little bit about the membranes and have a clear concept of the lipid chapter. Okay, and if you want to clarify all these chapters, you can enroll as a continuous student, and then we can solve all your queries related to online. Okay, and now let us move to the question 37. This is asking about the copy number of plasmid in bacterial cells that can be determined by, they are talking not about the copy number of the plasmid, other than they are asking how they can be determined. They are asking about the techniques, right? They are asking about the techniques, right? So what techniques can we apply to increase the copy number of the plasmid? You know, I should discuss some detail of the plasmid. This is the bacterial cells. These bacterial cells, they have plasmid, circular plasmid, right? Extracellular plasmid is there, and they are kind of DNA, right? Free in the cytoplasm. They have no nucleus. So their copy number, they divide and they can increase their copy number. This is called as the copy number. Right, so how you can determine about the number of copies, number of DNA content will be higher, and you can determine easily about the number of copies. So let us watch Western blotting, Southern blotting, Northern blotting, and quantitative PCR is given. So plasmid DNA, quantitative PCR you can perform. Western blotting is related to protein. This will not be going to much helpful. Southern blotting is related to DNA, RNA, another blotting is related to RNA. So there is no question of protein RNA. So option B and D will be correct. Now moving to question number 38, you have to choose the correct statement about a nucleosome core particle. They are asking about the organization of the DNA in the eukaryotic DNA, right? They are asking about the packaging, because nucleosome is a packaging particle where DNA is packaged. DNA is packaged tightly. So what they are asking about, which statement is correct? Out of 4, there may be one option may be correct, one option, more than one option will be correct. So they are asking about packaging unit of eukaryotic DNA. Yes, it contains 8 histone of 4 types. Yes, because you know H1, H2A, H2B, H3 and H4. So they are the twice. 1, 2, 3, 4. H1 is the sealing molecule, so this will not be correct. H2A, H2B, H3 and H4. They are occurring twice. There are 8 histone of 4 types. Yes, solenoid, all the DNA are packaged like this in solenoid form. This is the solenoid form. This is the solenoid form. It contains 8 histones. No, there is only one histone, that is H1. That's why C is wrong. Histone can be dissociated from nucleosome using high salt concentration. Yes, because whatever the histones are attached, they contain certain positive amino acid, right? As soon as you are using the high salt concentration, for example, you are using high sodium ion, then there will be high repulsion, alright, due to high repulsion, but they can be separated because bond will be weaker. Bond, the associated bond is there, that is binding those histone and DNA nucleosome, so bond will be finally weaker. Okay, so that's correct. Histone octamer, H2, H2B, H3, H4 we have discussed. They forming assemble into octameric disc. All 4 histone proteins share a high similar structure motif. The histone fold comprising 3 alpha helix connected by 2 loops, alpha 1 loop 1, alpha 2 loop 2, alpha 3. So histone octamers consist of 4 histone heterodimers, 2 of each, H3, H4, H2A, H2B. The histone fold motif of heterodimers are arranged with loop 1 of one monomer closely juxtaposed to the loop 2 of the second monomer, shown here for H3, H4 heterodimers, and axis of symmetry pass between the 2 long alpha 2 helix of 2 monomer. H3, H4 heterodimers pairs to form tetramer through interaction of 4 helix between alpha 2, alpha 3 of H3 form the dimer association of H3, H4 tetramer DNA, the first step in the nucleosome. So this is H2, H2 heterodimer bind to H3, H4 to tetramer via another homologous 4 helix bundle alpha 2, alpha 3 from both H2B and H4 joining the H2B, H4 histone folds. So basically we can conclude that they are octamers, right? So moving to question number 39, they are asking about what is bacterial superantigen and what is correct about them, right? So bacterial superantigen, LPS lipopolysaccharide is a special molecule over the membrane of microbes. They are acting as the antigens. Whenever we are talking about the superantigens, means they are activating certain special kind of immune cells. That's why they are called the superantigens. If you want to learn more about immunology, in-depth immunology, you can contact us. We can give you the exclusive classes regarding this immunology. So you can register here further on the website, and we can give you all these lectures. So what is we are discussing about superantigens. So let us see the options. The option A is given bind to BBC CDR2 loop in T cell without being processed into peptides, bind to BBC CDR2 loop in T cell after being processed into peptide. They are focusing especially on the processing. And the third one is recognized by B cell after being processed in peptide. ये ठीक नहीं लग रहा है. And about the fourth option, bind to BBCDR1 and HB4 loop in T cell without being processed into. Let us see which option is correct. So 2 option is correct. In the option A and D is going to be correct. This because this is not related to the B cells, that's why this is wrong. And bind to BBCDR2 loop in T cell being processed into peptide, this is wrong. Alright, so this is going to be work without being processed, and also bind to BBCDR1 as well as bind to BBCDR2 in the T cells, because B cell mediation is not there. That's they are helpful in activation of the T cells. That's why we are calling them as the superantigens. And superantigens are a class of special antigen that cause non-specific activation of T cells. As a result, there is activation of polyclonal T cell activation and massive cytokine release. And these superantigens are produced by some pathogenic viruses, HIV, and bacterial membrane, most likely as a defense mechanism against the immune system. You must read it well. And the superantigen bind first to MSC class second molecule. Remember this is very important because there are two classes, MSC molecule, MSC class 1, MSC class 2. So they are operating via, if you will see, superantigens. You can write in the short like SAGS. They are operating via this pathway. They are operating via superantigen, MSC class 2, and then coordinate with the alpha, variable alpha or beta chain on the TCR. This is the T cells, right? This is the T cells. This is around T cells. They are non-specifically activating non-specifically. Please remember, because always non-specific activation. Because always there is a specific activation of T cells, right? Whenever their antigen is involved. And what is the difference between antigen and superantigen? Antigen interacting via this MSC1, right? This MSC1, while superantigen is interacting via MSC2. And non-specifically, this is specific activation, right? This is specific activation of T cell after fragmentation presents MSC1 cells. But superantigen has been presented by MSC2 and they are non-specific T cell. That's why this mechanism is available in HIV, malicious viruses, and some bacteria, right? So here difficulty level is 4, more than 4 you can see, because you have to know about the MSC, you have to know about the activation, all the mechanism related to the T cell activation. T cell activation you must know, then only you can solve. So immunology is one of the most important part. You can see various questions are framed from this, right? So one example I can give you. Bacterial superantigens are staphylococcal enterotoxin A, termed as SCA, and second one is the B, termed as SCB, that mimic nominal antigens in that they are present in the MSC class second molecular TCR. Superantigens do not need intracellular processing and activate T cell bearing particular TCR beta region regardless of their antigens. Similar results have been found with rabies, cytomegalovirus, and HIV. So now let us solve question 40. When grown in presence of glucose and lactose together, E. coli does not utilize lactose first. Alright, so if this is talking about when grown in presence of glucose and lactose together, E. coli, they are talking about the operon, and they, right, so you have to select which one may be correct. You can see the lactose permease is not present, this is wrong. Glucose inhibits the synthesis of cyclic MP, this is right. And glucose activates synthesis of cyclic MP, very good, yes, this is correct. But glucose stimulates the efflux of cyclic MP, this is wrong, because glucose is not a transporter protein. So you need a transporter protein to pump cyclic AMP, to pump only the transporter protein can pump cyclic AMP out of the cell. So glucose is not a, this is, that's why this is wrong. Lactose permease is the part of this transporter protein. They are always present in the bacterial membrane. So let us continue. So this is about the lycopyron. I should remind you a little bit of this lycopyron, how they are working. This is a promoter site, promoter attached to promoter. On the downstream side, this is the downstream side. This is the downstream, and this is the upstream, right? This is upstream, lag Z, lag Y, lag A, right? So whenever glucose is available, right, when low glucose is there, what happens? When low glucose are there, cyclic MP is produced. What is cyclic MP? ATP hydrolyzed to form a molecule called as the cyclic AMP, right? Adenyl cyclase enzyme. This is here, adenyl cyclase enzyme is involved in the formation of cyclic MP. Whenever the glucose is low, what happens? This cyclic MP attaches to a regulator protein called as the cap. Both binding are there, right, at low glucose. As soon as there is a low glucose. Now what happening? This cap helps RNA polymerase bind to promoter. So now cap is occupied. The cap is occupied. Alright, cap is occupied by the cyclic MP. Now cap helps RNA polymerase to bind and resulting in high level of transcription. So now when high glucose is there and no cyclic MP is made, cyclic MP cannot bind to DNA without cyclic MP. So transcription occurs only at low level. Now, so cap is going to block. If cap is free, they will block RNA polymerase binding site. RNA polymerase binding site. So what will happen? So if in this case cap is not free, so cap will not be able to stop the transcription. Now they can block or stop the transcription. That's why you can see there is a high transcription in the case of low glucose. Okay, so now we'll solve 41 to 50 questions that are numerical type. Some of mathematical I will leave. And that will be solved by your mathematics teachers, and whatever related to biology I am surely going to solve. Let us talk about the question number 41. A plasmid digested with a restriction enzyme to produce 3 fragments of size 7 kb, 3 kb, 2 kb, and to obtain 500 nanogram of 3 kb fragment, the amount of plasmid required to be digested. This is the question, level 4 difficulty level. So after calculation, the answer you will get around 1.9 to 2.1, and you can see 3 kb fragment. You can write 3 kb 500 nanogram of a 3 kb fragment, and there are 3 fragments, 7, 3 and 2. This is a 3 fragment. Now they are especially focusing on 3 kb fragment. This can be written as 3000. Kb means kilo. So 3000, and that's multiplied by its molecular weight. Molecular weight is 660, and now you can get around 1.9 into 10 to the power 6 plasmid fragments. Okay, so now they are asking the amount of plasmid required to be digested in mg is, right? So DNA molecular weight is given 660. So you can multiply to obtain this quantity, and they must be divided by 10 to the power 6 to obtain that number. So what about the question number 42? A pentapeptide consisting of amino acid with molecular weight of 165, 131, 75, 204, 146 Dalton has an approximate molecular Dalton of कितना molecular weight होगा? How much molecular weight on there? If there is a pentapeptide that consists of various amino acids 165, 131, 75, 204, 1. So since peptide bond formation is condensation reaction, so what you have to do? Amino acid 1, then amino acid 2, amino acid 3, amino acid 4, amino acid 5. 1, 2, 3, 4, 5. So one molecule of H2 second molecule, third molecule, fourth molecule, 5. So 5s2 molecule. Alright. Sorry, 1, 2, 3, 4, only 4. Only 4 molecule of H2 will be released for the formation of pentapeptide. Okay, pentapeptide. Now you have to add all these molecular weight and then subtract by the molecular weight of water. Then you will get this range. So I think it is clear to you. Now the next question, number 43. At 2 mg per ml pure tubulin concentration, a microtubule consisting of 13 protofilament grows unidirectionally at a rate of about 2 micrometer per minute. At this growth rate, number of tubulin dimers, each 8 nanometer in length, added to the ends of microtubule each second is, right? So what they are asking, at this growth rate, the number of tubulin dimer, they are asking about the number of tubulin dimers that will be added at the end of microtubule. So you must know that rate is a length upon time. Length is given 2 micrometer per minute. So length is about 2 micrometer. 2 micrometer, you can write is a 2000 nanometer. Alright, nanometer per second. You can see they are asking the answer in nanometer, I think. Okay, number 2, each 8 nanometer in length. Alright, length is in nanometer. So per second, so 2 micrometer. 2 micrometer, you can write as a 2000 nanometer, right? Nanometer divided by this per minute. Per minute, you can divide by 60 to get second. Now you will get this much of. So they are asking about the number of tubulin dimer each 8 millimeter. Each 8 millimeter. So this is the rate of growth per second. But you should divide 8 nanometer in length, that is the 33.3 multiplied by 13. Why 13? Because there is a 13 protofilament, and then you should divide number of tubulin dimer, each is 8 nanometer. Then you can accurately tell how much the actual number of tubulin dimer can be added per second. This will be around 54. So this is solved. Now what about the 44? It's a easy level. 3 sides of triangle are 1.5 meter in length. The area of triangle in meter square. So you can calculate it by yourself. Sum of the internal angles of regular pentagon. You must remember this angle because there is a fixed angle pattern. You can solve 46. A protein solution gives observance of 0.34 at 280 using 0.5 centimeter path length. Given the extension coefficient of a protein is 3.4 into 10 power 3 meter per centimeter, the concentration of protein in the solution in millimeter is. So you can solve by using the formula, absorbance upon path length into extension coefficient, and you will get this much of the answer. Okay, so in question number 47 they are asking about the pitch of an helix is around 5.4 angstrom. The approximate length of a helix consisting of 15 amino acids in angstrom is. So you know pitch has given, this is 5.4, and approximate length of helix consisting of 15 amino acids in angstrom they are asking, right? So you have to calculate per turn. You can divide by 3.6 and it will be around 1.5 upon 1 residue, means length. This is the length 115. You can multiply by 1.5. Alright, then you can get the actual length and it will be around 22. I think this is easier one. And question number 48. Maximum resolution in nanometer that can be achieved through an oil immersion lens of numerical aperture 1.4 using an incident light wavelength 380 nanometer is. So you must remember this formula of resolution. Resolution is 1.22 lambda upon 2 into numerical aperture. So replacing a value, you can directly calculate this maximum resolution. So they are asking about, this is memory type question, question number 49, number of NADH molecule required for synthesis of formatted. Formatted has carbon number 16 from acetyl coenzyme is A. So number of NADH molecule they are asking, not ATP, please remember that. So the number of peptide fragments generated from sequence, the alanine, tryptophan, valine, alanine, valine, threonine, glycine, lysine, glycine, serine, lysine, and treatment with chymotrypsin is, chymotrypsin actually hydrolyzes only the aromatic amino acid. So you have to mark where is the aromatic amino acid, for example this, right, tyrosine, right, so phenylalanine, tyrosine and tryptophan, you have to look for this, the tryptophan. So it will be around 3 to 4 number of peptide fragment that can be generated. So now question number 51 to 52 marks each, and you have to guess the answer because there is no option, right, and also there is no negative marking in these sections. So they are asking about the prevalence of a severe form of sickle cell anemia in African population 16%. The percentage of the population resistant to malaria because of the heterozygous genotype for sickle cell gene is, right. Sickle cell anemia occur in the recessive form, and they are giving it's a 16%, right. Now they are asking a percentage of population resistant to malaria because heterozygous condition. They are asking about the heterozygous. You have to apply P plus Q equal to square means P square plus 2PQ plus Q square. Q square they are giving the 16%. Now they are asking about the 2PQ. You have to calculate the P, right. So it will be around 47, 49. Let us see. So for 16% you can calculate this Q square. Q square will be 0.4 now. You have to find out the P. You can subtract because P plus Q is equal to 1. So you can calculate P by 1 minus 0.4. It will be 0.6. Now they are asking about the 2PQ, which will be 2 into 0.6 into 0.4. It will be around 0.48. So you can solve it. It will be around 42. What is the maximum velocity of an enzymatic reaction? Is 0.4 mole per second at 5 mm concentration of the substrate. The reaction velocity is found to be 0.2 mole per second. If the enzyme shows standard Michaelis-Menten kinetics, the rate of the reaction at 10 state concentration will be. This is from the enzymatic kinetics. So you must remember that rate is equal to 1 by V max into 1 by S. So directly you can put the value and you can calculate. So what about this question 53? A chromatin fiber of 14 nanometer length contains 25 nucleosomes, around 200 base pair per nucleosome. The degree of compaction of DNA associated with this chromatin fiber is how much fold? So this is given 2540 nanometer length, contain 25 nucleosomes, and each nucleosome is containing 200 base pair per nucleosome. So they are asking what the degree of compaction of DNA associated with this chromatin fiber. So how you can solve it? You can solve it by multiplying. The number of nucleosomes is 25 and each compaction is around 1.6. So you can calculate the degree of compaction around 42.5. So each of the histone, since all the histone come together to form a histone octamer, which binds and wraps approximately 1.7 tons of DNA or about 146 base pairs. So this 1.7 must be multiplied by this 15. Then you can get your answer. So this question is related to mathematics. The ball is dropped from a height of 1 meter. Every time the ball bounces up, it reaches 50% the height of previous bounce. It rises up to 0.5 meter on the first bounce, 0.25 second bounce, and after infinitely long time total. My mathematician teacher will solve it. So I'm leaving for that. This is also related to the mathematics. And also I'm leaving this question for our mathematics teachers to be solved. You can see 57 is also related to the probability question. They are asking an unbiased coin is tossed 4 times. Probability of getting exactly 2 head and 2 tails in any order. What will be 4 times? So 4 into 3 into 2 into 1. Alright, so they will solve. Now, since this is biological problem, I am going to solve it. Question 58. Rod set equali is 2 nanometer long and has diameter of 0.8 millimeter. The average density of equali is around 1.1 into 10 power 3 gram per liter. The mass of single equali cell in picogram will be. They are asking the mass. So first you should calculate. This length is given. Length is around 2 nanometer long, means 2 into 10 power 6. Radius is given 0.4, right? Radius, because diameter is 0.8, so you must divide by 2. It is 0.4, and density is around 0.44 millimeter. You can convert them into the further. Okay, so they are asking about mass. So mass is equal to volume into density, and volume is pi r square into density. Now you can solve it and you will get the answer of 12.2. So 59, the length of each side of regular octagon into area of octagon in meters square is. So my mathematics teacher request them to solve and post a video on this separately, so you can look for that answers. So this is the last question. Molecular weight of genomic DNA of an organism is 3.96 into per mole. The average molecular weight of nucleotide pair is given. If an average protein in this RNA is chain 400 amino acids, the maximum number of protein by DNA coded by DNA molecule will be. So the number of nucleotide pair DNA is given. You can divide by 660 gram per mole and you will get this number, and 1/3 of codon will be, because total number of codon has been given, I think 6 into 10 power 5 you have obtained, right? So it should be divided by 3. You will get the total number. This will be the nucleotide, and total protein coded by 2 into 10 power 5 upon the number of 400 amino acids. So you can calculate it around 5,000. Thank you. Please visit more. If you have any feedback and a request, you can post your request. We will try to give you the exact solution. Thank you. Bye bye.